In clinical medicine, a few hours can dictate a patient's survival, but isolating a single protein and mapping its exact structure using traditional methods like X-ray crystallography can take months of painstaking chemical preparation. The modern physical alternative skips the purification entirely. We can take a single drop of human pleural fluid straight from the patient's lung cavity and place it directly onto an optical sensor. This process relies on Fourier transform infrared spectroscopy, or FTIR. FTIR is an analytical technique that uses infrared light to measure how molecules absorb energy, allowing scientists to study proteins in their native environment without complex sample preparation. This graph shows the immediate problem with raw FTIR data. Because the fluid contains thousands of overlapping biological molecules, the output is a chaotic superimposition of structural noise, where the actual biological signal is buried. The clinical objective hidden in that noise is highly specific. We need to distinguish the chemical markers of malignant pleural mesothelioma from a harmless, benign transudate. The speed of the optical sensor offers no clinical value if we cannot mathematically extract the exact chemical signature of cancer from the surrounding noise. Standard human observation or simple statistical curve fitting fail when confronted with this volume of overlapping spectral points. Solving this requires a custom computational pipeline built at the intersection of three specific disciplines: experimental biophysics, applied mathematics, and artificial intelligence. This diagram outlines the required architecture. The process starts with physical feature extraction of the raw spectrum, moves through a mathematical filter for data reduction, and ends in the topology of an artificial neural network. An artificial neural network is a machine learning model inspired by the structure of the brain that recognizes complex patterns and numerical data. These three stages operate in a fixed sequence, translating complex physical matter into a digital classification. Constructing this specific pipeline allows us to recognize diagnostic patterns invisible to the human eye, solving the bottleneck of rapid clinical diagnostics. The first step in this pipeline requires analyzing the raw FTIR spectrum to locate the physical signature that defines a protein's secondary structure. This chart shows the raw spectrum. Most frequencies fade away, isolating one specific region, the amide I band, located between 1600 and 1700 inverse centimeters. This band is generated by a precise physical phenomenon: carbon-oxygen bonds stretching and vibrating along the protein's peptide backbone. The exact frequency of these microscopic vibrations shifts depending on how the protein is folded, dictating whether it forms an alpha helix, a beta sheet, or remains entirely unstructured. Traditionally, human operators attempt to read this band using manual deconvolution methods, but those techniques are highly subjective and easily distorted by background noise. Isolating the amide 1 band physically provides the raw material, but we still need an objective mathematical translation before any artificial intelligence can process it. The immediate temptation is to take these newly isolated amide 1 spectral points and feed them directly into the neural network. However, biophysics suffers from a strict small data bottleneck. The ground truth database required to train the network contains exactly 35 known protein structures. As this diagram demonstrates, pushing thousands of highly dimensional spectral points through a deep learning model trained on only 35 subjects causes the network to memorize the noise instead of the pattern, leading to a failure state known as catastrophic overfitting. To breach this gap, the data must pass through a specific mathematical filter called the discrete wavelet transform, or DWT. The DWT is a mathematical tool that decomposes a signal into mutually orthogonal sets of wavelets to capture both frequency and location information simultaneously. The discrete wavelet transform functions as a data reduction technique, distilling the raw signal into its most essential components. This animation shows the mechanics of the transform, breaking the smooth curve apart into a series of smaller, staggered, and localized wavelets by applying mathematical scaling and shifting. This matrix reduces the data size while retaining the structural geometries of the original protein. By shrinking thousands of data points into a dense mathematical summary, the wavelet transform bridges the gap between high-dimensional physical data and the strict input limits of small dataset machine learning. With the data compressed, we move to the final machine learning phase, feeding the wavelet matrix into the input layer of the artificial neural network. Because the dataset is limited to exactly 35 proteins, standard training protocols, like an 80-20 split between training and testing data, are mathematically insufficient. This 35 profile grid illustrates leave one out cross-validation. The network uses 34 profiles to train its parameters, withholding exactly one to test its accuracy. It then systematically rotates that single test subject through every profile in the grid, ensuring robust learning across the entire limited dataset. By repeatedly testing itself against a rotating, unseen profile, the network successfully learns to generalize the protein structures without overfitting the data. The results of this architecture are highly precise. The model achieved a 96.88% prediction success rate in identifying beta sheets and a 93.92% success rate for alpha helices. Hitting these exact accuracy thresholds on known proteins proves the network is structurally sound and mathematically ready to process unknown clinical applications. We can now shift focus from the controlled training environment back to the real-world clinical targets from the beginning of our process. The system is deployed to evaluate 3 distinct human fluid pathologies: a harmless, benign transudate, standard lung cancer, and malignant pleural mesothelioma. Operating at speed, the raw fluid hits the ATR FTIR sensor. The amide 1 band is extracted, and the wavelet transform compression occurs instantly. ATR FTIR stands for Attenuated Total Refractome's Fourier Transform Infrared Spectroscopy, a technique that allows samples to be examined directly in the liquid state without further preparation. In the final stage, the trained neural network processes those coefficients to identify significant, measurable differences in the specific protein content of the 3 fluids. This 3D classification plot reveals the final output. The previously chaotic data cleanly separates the benign transudate, lung cancer, and mesothelioma markers into 3 distinct color-coded spatial clusters. This clear spatial clustering proves the system can differentiate an asbestos-induced cancer from a benign fluid based purely on the mathematical patterns hidden within the protein's secondary structure. The careful synthesis of optical spectroscopy, rigorous signal processing, and specialized AI successfully transforms raw biophysical noise into actionable, life-saving diagnostic data.